Biological specimen. Biological specimens. Blood is the most common biological fluid collected from clinical laboratory testing. It's usually drawn from vein in the arm directly into the evacuated tube. Directly into an evacuated tube. Blood is the most common biological fluid collected from clinical laboratory testing. It's usually drawn from vein in the arm directly into the evacuated tube. Typically, a tube will hold about 5 ml of blood, enough to perform many clinical chemistry tests, since automated analyzers require only small amounts, usually from 2 to 100 microliter for a single test. Occasionally, when collection of the blood from a vein is difficult, a sample of capillary blood may be collected by breaking the skin and collecting several drops of blood from the puncture site. An example is the use of heel stick blood from testing of newborn. Here, phlebotomy, the act of drawing a blood sample from blood vessel. For clinical chemistry testing, blood is usually drawn from a vein, typically a vein in the arm or back of the hand. Collecting blood from a vein is called venipuncture. The medical professional drawing the blood sample is called a phlebotomist. Phlebotomy is the practice of drawing blood from patients and taking the blood specimen to the laboratory to prepare to testing and researches. Other biological fluid matrices often used for testing include urine, saliva, cerebrospinal fluid, amniotic fluid, synovial fluid, pleural fluid, peritoneal fluid, precardial fluid. These fluids often contain the same biological analytes for interest such as glucose and protein, but differ greatly from each other in physical and chemical properties. These differences in fluid characteristics are termed matrix differences. Test methods that are designed for determination of an analyte in the blood plasma may not be suitable for determination of that same analyte in the other matrices. When using a test method for analysis of a fluid other than blood, plasma, or serum, it's important to validate that the method is acceptable for the type of fluid sample being used. Fluid typically used for clinical chemistry tests: blood, whole blood, serum or plasma, urine, cerebrospinal fluid, amniotic fluid, saliva, synovial fluid, fluid that is found in joint cavities, pleural fluid from the sac surrounding the lungs, pericardial fluid from the sac surrounding the heart, peritoneal fluid, also called acidic fluid from the abdomen. Blood is the most commonly used specimen for testing in the clinical laboratory. Blood consists of two main parts: a fluid portion called plasma, which contains dissolved iron in the globule, and a cellular portion, the red blood cells, white blood cells, platelets. Most clinical chemistry analytes are found in the plasma. Part of the preparation of blood for testing these analytes involve removing the cells. This is done by centrifugation of the sample to pack the blood cell in the bottom of the collection tube and allow removal of the liquid portion of testing. Here, blood collection, centrifuge, we separate plasma from blood cells and clotting protein. Here, blood collection tube with no anticoagulant remain for 30 minutes. Clot of care, centrifuge, we get serum, cell plus protein clot. Preparation of serum or plasma. If the blood sample is collected in a tube containing an additional that prevents the blood from clotting, called an anticoagulant, the fluid portion of the blood is called plasma. If the blood is collected in a tube with no anticoagulant, the blood will form a clot. The result liquid above the cell and clot is called serum. Serum contains all the components of plasma except the collecting protein, which are consumed in the cascade of reactions that form the blood clot. Some clinical chemistry tests are best performed using plasma, others are best performed using serum, and still others can be performed using either plasma or serum. Tubes used to collect blood have color-coded caps that signal what, if not additives, are present in the tube. Additive may be anticoagulant to allow preparation of plasma, or may be substance included to protect annelids from chemical or metabolic breakdown. Certain types of anticoagulants may be incompatible with some kind of test. For example, Edita is an anticoagulant that inhibits the clotting of blood by sequestering calcium ions that are necessary component of clotting reaction. However, samples of plasma collected using Edita tube are generally unsuitable for measurement of calcium and for any test method that involve a reaction step depending on availability of calcium. Here is the tube, clot activator tube. Types of blood collecting tubes commonly used for chemistry tests: tube additives and tubercular sampling and comment. The tube additive plant, tubercular red, sample serum, common. Clotting required at least 30 minutes at room temperature. Tube silicate color activator, red or black. Serum silica speed the clotting process convert to an activator. Thrombin tubercular green. Serum thrombin speed clotting process significantly produce serum in several minutes, used mostly for urgent state testing. Green isthmus heparin. Isthmus heparin green requires plasma sample for most chemistry tests, not suitable when testing for lithium. Sodium heparin green is not suitable when used for testing for lithium, not suitable when testing for tube additive Edita, tubercular lavender, sample plasma, used for hematology. Edita lavender, tubercular lavender, potassium Edita, Dan Brown for blood lead testing tube, very low level of contamination by lead. Sodium fluoride, potassium oxalate, gray. For glucose testing, sodium fluoride inhibits metabolism of glucose by white blood cells. Some collection tubes also contain an inert silica gel that positions itself between the cell and the serum or plasma during the centrifugation step. It seals the cells in the bottom of the tube and prevents substances that leak from the throat from contamination the serum or plasma. These are called serum separator tube, designated SST, and plasma separator tube.